Glioblastoma, which is the grade 4 glioma or brain cancer, is the deadliest form of brain cancer. So the average survival is about 13 months after diagnosis, and that's with very aggressive treatment. And often patients come in with a diagnosis because they're having severe neurological deficits. So there's kind of the standard of care therapy that is put into place because you have to remediate those bad side effects.
We've, using a collection of different experiments, have identified a target that's expressed specifically on the glial cells around the tumor that's upregulated by interstitial fluid flow. And part of the grant is looking at targeting that molecule and reducing the effects of the glial cells back onto cancer cell invasion.
We'll take the MRI images that we get from University of Virginia and develop the code and the algorithms mathematically so that we can describe the fluid flow and fluid movement. We'll analyze tissue samples, and we'll use these cells to actually do things like live cellular imaging in 3D engineered microenvironments. We will develop new systems in which we can kind of image these cells, analyze these cells, and kind of really the bulk of the work will happen here. We really are working with, we're working with other engineers, we're working with like MRI physicists, we're working with physician scientists, oncologists, as well as basic scientists and chemists to really look at something that's totally unexplored. The end goal is that we identify therapies that can be used against glioma. And really by looking at interstitial fluid flow and fluid drainage in brain cancer, it's a completely untapped area to look for targeted therapies. So we're really taking everything that's out there and looking at it in a new context. And the hope is that we can find something that's uniquely able to stop brain cancer invasion and assist the standard of care that can actually cure patients.